Hello and welcome to another segment of Interviews That Matter. I'm your host Raj Mehta. Friends, in this segment we bring those guests who influence our lives. This includes elected officials, policymakers, heads of major organizations and other dignitaries. It is my sincere hope that the knowledge brought in by these guests will help our community. Today we have a different type of guest. He is Dr. Atwal. He is a scientist and does a lot of research on human genes at Cold Spring Harbor Lab. Mickey, thank you so much for coming.
That was a real pleasure.
Taking time from Manhattan, coming from Manhattan in a train and a taxi. Not bad, right?
Yeah, it was pretty easy. Yeah.
Pretty easy.
Yeah.
Pretty, okay, good. So, we always begin with your, background, basically. Like I know you're a scientist, but for the viewers' benefit, how did you become scientist, or what is your background? Where did you?
Sure.
So
okay.
let's go over the history.
Okay. Wow. So in my previous life, like every good boy who has parents of Indian descent, I went to medical school.
Okay.
At the University of Cambridge. But like every bad boy, I quit. And the reason I quit was because I really hated it. My real love was mathematics and physics.
Okay.
I grew up watching documentaries about science. I read the books of Carl Sagan, Stephen Hawking. I love solving math puzzles. So that was really my forte. And after a year or two, I just dropped medical school and went into the physics program.
Oh, so you dropped out?
I dropped out, pursued a bachelor's in physics, a master's in physics, and then this is still back in England. Mm-hmm. And then I went across the pond to the United States to pursue graduate studies, did a PhD in theoretical physics. Mm-hmm. And I found myself slowly working my way back into biology. So my mum is kind of now slightly happier. I'm not a practicing clinical doctor, but it sounds similar.
It's
right?
similar.
Yeah, I'm working in a similar field. I still get to call myself a doctor, but not a real, not a practicing doctor. So after my PhD in theoretical physics up at Cornell, I pursued postdoctoral studies in New Jersey at Princeton, mm-hmm. and that's when I started getting seduced into biology again. Mm-hmm. And the reason for this is because around about that time, in the early 2000s, biology had undergone a revolution. Mm-hmm. It had really become more quantitative mm-hmm. in a way that it wasn't before. Mm-hmm. If you look at the textbooks back in the 80s and 90s, they were very qualitative in biology. Mm-hmm. Nowadays, there's been a revolution whereby there's been a lot of data being generated, and so there's been a real need to apply the tools from mathematics, computational science, and physics into biological problems. Mm-hmm. So after that stint into working on biological problems, I became seduced, and I realized this is my calling, and I'm now working as an assistant professor at Cold Spring Harbor Laboratory, mm-hmm. not too far from here.
Wow, okay, great. And your parents are from India?
Yeah, my father was born in Punjab, in mm-hmm. a small little village, mm-hmm. In the Indian side of the border, and mm-hmm. my mum was born on the Pakistan side of the border. Oh. So this is pre-partition. She was born in Lahore.
Oh, okay.
And they got married in England, and I was born.
Oh, okay. So you were born in England, and
yes.
you grew up in England.
Yeah, well, I was born in England. I decided after a few months I really hated the weather, and so I went to Iran.
Oh, interesting.
Well, my father had a job over there.
Oh,
so
okay.
I lived in Iran for a few years. Fantastic, I have to say. One of the best years of my life. I mean, it always is. Your childhood years are always the best years of your life.
Right.
But this is pre-revolution. So the Iran back then, or my memories, it's very different from the Iran nowadays. So after the revolution, we were essentially evacuated by the British embassy. And we spent some years living in different parts of the world, like Kuwait, Syria, India, and eventually I came back to England.
So your father was like working for government, or?
No, he worked for the oil industry.
Oh, he worked for the oil industry.
Yeah.
Oh, okay. That's how he got to move around all these places, right?
Right.
And took you everywhere. And,
yeah.
And your experience was the best in Iran?
Only because
or because you were growing up and you were a child?
I was worry-free
okay.
as a child, right? There's no school, nothing, just going out to play every day. So that was a lot of fun. But my formative educational years were really back in England. So that's where I went to high school and undergraduate.
Okay, okay. So, you know, I was actually I attended one of your lecture at the Long Island Forum for Technology, which was really great, and I really learned a lot out of that, and we want to thank you for that. So the question of big data. First, let's talk about big data.
Okay.
What is big data? I mean, you keep on hearing about it in the paper, in newspaper. You keep hearing on the TV and everywhere. What is big data?
Yeah, it's a big buzzword that's thrown about. I mean, I have to confess, I do feel kind of dirty using that phrase because it's kind of overused. I feel it's in the peak of the hype cycle. You know, every time there's a hype about something new, there's a bubble and there's a burst because people expect too much out of this new idea. So there's always been big data, right? There's always been far more data than we can analyze by humans. So there's always been a need for computation to analyze data. And this is certainly true in commerce, in finance. What's happened in biology is we've now been able to sequence and analyze DNA. And now DNA, as you might remember from high school, is a very long molecule that's found in every cell. I mean, if you were to stretch it out, it'll be 2 meters long. Right? And the DNA contains roughly 20,000 genes. And these 20,000 genes essentially are the blueprint that characterizes why you are different from me. These genes tell you, what skin color you have, what eye color you have. They determine how you metabolize certain foods, and they're really responsible for every functioning process in every cell. But it's a very long molecule, and it's broken up into essentially 3 billion chemical units.
Wow.
And so to understand our entire collection of genes, that's known as a genome.
Right.
To understand the entire collection of genes, we have to know what that exact order of 3 billion units are. So that's a lot of data.
3 billion?
3 billion for every person.
Wow.
Right?
Every person?
Every person, right? And I mean, every person has a unique set of 3 billion units.
In sequence, you mean?
Exactly.
I have a different sequence of 3 billion versus you.
Exactly, right. So you have 2 copies of DNA. You've inherited one from your father and one from your mother. But those copies aren't perfect copies. There's always some errors, and these errors we call mutations. It's a reason why you don't look like, this is a reason why people look different. Right. This mutation is a good thing because it allows genetic diversity to appear in a population. And some of these mutations can be very beneficial. A good example is there's a mutation that arose roughly about 10,000 years ago, somewhere in Eastern Europe. And those individuals who inherited that mutation can drink milk. They're lactose tolerant. And those individuals who can't drink milk don't have that mutation after the age of 2. So these errors in your inherited copy of DNA can sometimes be a good thing, but sometimes they can be a very bad thing, and they can lead to early onset of certain diseases. So a big part of biological research today is to understand how does the sequence of DNA influence our health in the future. So it's kind of like a magic window in time. In terms of science, we're generating a lot of data, but we really don't understand how to interpret or translate this data. And the other useful aspect of understanding this is that it allows us to understand our genealogy. So you've inherited your DNA from your parents. Right. Your parents inherited their DNA from their grandparents. So the patterns and mutations contain an imprint of your past. Your ancestral past. So by looking at those patterns and mutations, we can infer something like, where did your ancestors come from? Recently, and which wave of migration out of Africa did your ancestors come out on? So it's a really nice window into the past, and it's also a small window into the future, because hopefully in the future, and this is the promise of medicine in the future, by looking at those patterns of mutations, we can predict, with some certainty, who's likely to, suffer certain ailments compared to others, and maybe we can do something about this beforehand.
Interesting, interesting. So that is your research right now you're doing on at the Cold Spring Harbor Lab.
Right. So I work in the quantitative biology program.
Okay.
So quantitative biology is really a new field in biology. It wasn't around when I was studying medical school. Otherwise, I would have stuck in medical school if they had, had lots of computation and mathematics. So it's a new field. It really sprung up 10 years ago, and it's brought in a lot of expertise from computational scientists, mathematicians, physicists, all applying their trade to understand this genetic code, interpret this code, dealing with these large amounts of data. And there's a lot of problems, computational problems we have to deal with. Like I've mentioned, there's a lot of data that you generate from a single person.
Single person, right?
Single person. So to sequence one person's DNA, you have to generate on the order of 100 gigabytes of memory, of information.
100 gigabytes.
100 gigabytes. So just to put that into perspective, a book, the size of a typical large book, if you just look at the word content, you can compress that information into about a few megabytes.
Right.
So 100 gigabytes is the information content in a stack of books about 6 miles high.
Gee.
Right.
Gee. For one person.
No, sorry. I'm taking it back. No. I'm taking it back. No. At Cold Spring Harbor, we generate a terabyte of data.
A terabyte of data.
Every day.
Every day.
Every day. That's 1,000 gigabytes.
Oh, wow.
And that's from human sequencing. And the information content of that is like a stack of books a few miles high.
Wow. Wow. So, I'm not too knowledgeable in this, right? Obviously, So I wanted to find out, okay, let's say, for example, if you have to analyze my genes.
Yeah.
What do you do to analyze? I mean, what, like you take blood or what I mean, I'm sorry to ask you this question.
No. So we can take blood, or we can take cells from any other part of the body. Essentially, what we do is we break down the cell membrane, we extract the DNA, and then the DNA is passed into what we call a DNA sequencing machine.
Okay.
And it's this machine which then reads out all 3 billion chemical units, the sequence. These sequencing machines are fairly expensive. I mean, the latest machines are of the order of $1 million. So, it's not quite a meritocracy in terms of, which countries can afford to do this. It's expensive. But they've really come a long way. The first time the human genome was sequenced, that took roughly 13 years, from 1990 to 2003. Just one individual.
Just for the one individual?
One individual, and that came at a cost of $3 billion.
Oh, wow.
Right, so that's certainly not something we could do, on a day-to-day basis for everybody, right? It's way too expensive.
Right.
And that was an amazing project. That was probably and still is the largest international biological collaboration. There were, it was spearheaded by the US and other countries like the UK, Spain, France, Germany, Japan, China, all joined in. They were essentially splitting the task and dividing the labor of that into different countries and different institutions. And what's happened since 2003 is that the ability to sequence DNA has changed dramatically. Right, with $3 billion and 13 years is way too expensive.
Yes. Yeah.
So whenever you have a problem, technology, there's always an impetus to change technology, drive technology to make things cheaper and faster and quicker.
Right.
And over the course of 10 years, the cost of sequencing the genome has gone down dramatically. It's been a huge precipitous drop. It's gone down from $3 billion to about $3,000.
Wow.
In the space of 10 years.
Wow.
I mean, that's a millionfold decrease.
That's
right.
a huge difference.
That's a huge. I mean, I can't think of any other industry where you have a millionfold improvement in the space of 10 years. Right. I mean, even in computing, right? There's something called Moore's law, where computing ability, processing ability improves every 18 months. It doubles every 18 months.
Right.
But this is faster than Moore's law.
Oh boy.
And so as of last year, it was $3,000, and there was an announcement by a company called Illumina in January of this year, and they have a new machine which is $1 million, and they claim that you can sequence a genome for $1,000.
$1,000.
$1,000, and it takes of the order of 3 days. As opposed to 3 days as opposed to 13 years. So that's got to be good.
Wow.
Right.
Wow. So basically the equipment is the one that analyzes everything and gives you a report, or how does it work?
Yeah, so at the end of the day, what we get is we get lots of fragments of that DNA.
Okay.
Chopped up, but that's how these machines work. They can't just read out every individual unit in a sequence.
Right.
They first chop up the DNA. The DNA is first chopped up, and then it reads these individual fragments, and then it's our job to piece those fragments together and reassemble.
So, okay, so the computer gives you fractions of information, and how many fractions? I mean, is there any kind of...
because we need to get very good overlaps between these fragments to get the entire sequence.
Right.
You want to resequence 30, 40, 50 times, which is why you generate 100 gigabytes of data. Because you have to resample and regenerate these fragments such that you can sufficiently piece together the entire sequence without ambiguity. It's still not perfect yet. There's lots of issues with the DNA sequence itself that makes it perfect, but we're getting close to that stage.
Is there like a manual process, or is it like something that you still go to the other equipment, you use other machines, other computers?
Oh, so this is
software,
where computational science has really saved the day.
Okay.
I mean, the problem of piecing together fragments of DNA into one long sequence, that's a computational science problem.
Right.
Right. You have 100 gigabytes of data.
Right.
You somehow have to be able to search through this, which is non-trivial. You have to then realign them together and find overlaps. And so there's been parallel developments in algorithms in computer science that have, people have come together and they've really made huge changes into how we interpret and how we sequence the DNA.
Wow. Okay. And at the end of the day, like for example, you said 3 days it takes, right, to analyze a DNA or put a sequence together, right?
Right.
And let's say if you take my DNA and it takes 3 days later, I know exactly who I am?
Well, it depends on well, you know what your sequence is.
You know what? Okay.
You know what your sequence is, but it doesn't tell you exactly who you are, because there are other factors which come into play. There's environmental factors, right? Your DNA contains all information to tell you how to recreate you. Your DNA tells you, that you should have 2 arms and 2 legs, and this is what's happening, during embryogenesis, when you're when you're undergoing development.
Right.
But if something happens to you, you lose an arm, that's nothing to do with your DNA. That's just, that's an environmental feature.
Right.
Or if you're exposed to certain pathogens, that's not necessarily, indication of, underlying DNA defect. That's an pathogen. So, so who you are is really a combination of 2 things. It's your DNA that you've inherited from mother and father, with mutations, and plus the environmental factors on top of this.
Okay, so like, I have 2 copies of DNA, right?
Yeah.
Within me, my mother and my father.
Yeah.
And let's say, for example, I'm asking you stupid questions, okay? Because
there's no stupid questions.
I'm stupid on this subject, basically. Let's say, for example, my mother and father has no issues in, let's say, their eyes are perfect, and still son or daughter has a bad eye. Is it like comparable? I mean, how does that work?
Okay, so not every disease is genetic.
Okay.
Right.
Okay.
For example, if you catch a cold,
right?
You catch influenza. That's not because of your underlying genetic predisposition. It may be, but that's not how what's generally thought to be at play. There are a catalogue of genetic diseases where we know that if you inherit a bad copy, there's a very high chance. If you inherit a bad gene, a bad copy of a gene, there's a very high chance that you'll get the same ailment as your parent who had that gene.
So birth defects are kind of part of that, or not really? Like, there are birth defects also, right?
Yeah, so there's a lot of congenital birth defects which are due to genetic abnormalities. And that could be due to either they've inherited bad DNA, or there's new mutations that have been introduced, in which have caused these defects. And the promise of medicine in the future is, because we now have a greater understanding that everybody's so different in terms of DNA, we really want to personalize medicine, right? The idea is that we don't want to have one drug that fits all. I mean, that's, you know, people have different responses to drugs, different, you know, different susceptibilities. And, you know, everybody has disease in a different way. For example, a big focus of my research is on cancer. Now, cancer is a genetic disorder. And why is that? Because what happens is you have a normal cell, and suddenly there are genetic changes that occur, and these genetic changes causes the DNA to be unstable, and these cells start proliferating in an uncontrolled manner. But these changes, these mutations, are essentially random. So if I were to develop a tumor, and I look at the DNA sequence of that, the changes that I would have would be very different from the possible changes that you would have if you had a tumor. So this dictates that the kind of therapy I would need would be very, very different from the kind of therapy that you would need. And right now, I mean, most disease treatment is essentially looking for, most research in disease treatment is essentially looking for a single silver bullet which treats everybody. But there's a growing appreciation that, you know, that doesn't work. There's a lot of people who don't respond the same way.
Exactly, right. Yeah,
and that's because of our genetic differences.
Oh, okay.
And so if we can understand what is it about our DNA differences that allows us to be more susceptible to different diseases, and what is it what is it about those changes that allows us to be more susceptible to different kinds of drugs, then maybe we can target and develop drugs that are tailored to an individual. So you can imagine a scenario in the future whereby a patient has an ailment, goes into a clinic, and again gets their DNA sequenced.
Wow.
Right.
Right.
And then the doctors are able to, sometime in the future, you know, using, combining that information with clinical data, then tailor a therapy for that patient. But we're still quite some time away from there. There's a lot of hurdles we have to overcome. A lot of computational hurdles. The biggest hurdle in big data isn't just the size of the data. The biggest hurdle is actually things to do with storage. Right.
Yeah, because
I can imagine, you know.
I mean, said 1 terabyte every day.
So
365 days.
Exactly. What do you mean? 1 terabyte a day. Imagine storing all information in a stack of books, you know, a few miles high.
Yeah.
Worldwide, there's 25 petabytes of data being generated. So a petabyte is 1,000 terabytes, and a terabyte is 1,000 gigabytes, right? So, you know, a movie that you might download might be 1 or 2 gigabytes.
Right.
So that gives you an idea of the scale. And that's tripling every year. Yeah, so there's a lot of data that needs to be stored. So that's one problem. And once you have the storage, you want to be able to collaborate with researchers or transmit that data. That's really inefficient right now. That's really slow. I mean, imagining transferring 1 million movies down the internet.
Wow.
It would take, you know, weeks and months.
Right. Yeah.
The biggest DNA sequencing facility in the world is the Beijing Genomics Institute in China.
In China, okay.
They generate so much data, you know, on a day-to-day basis that it's actually faster to transmit data not by internet, but by hard disk and FedExing the data.
Yeah.
Right.
Yeah.
So that's one issue. So they actually have to go back to an old-fashioned method. It's not quite horse and cart, but, you know, it's an analog way of transmitting the data. So that's one issue. You know, storage and transmission. The biggest bottleneck, I would say, is neither the size of the data, neither is the storage, it's neither the transmission, it's actually the interpretation.
Right.
We have no idea how to interpret most parts of the DNA. I say we have 20,000 genes, but we don't know what all those genes do. Right?
Oh, okay.
Right. And there's many regions of our DNA which aren't coding for genes, and we don't know what they do. Right. Presumably they play some role in switching on or switching off genes.
Right.
And that's going to have a huge impact on disease.
Right.
So So the cost may come down for sequencing DNA, but there's still an additional cost of being able to understand what that DNA means and interpreting it. So I would say the biggest bottleneck now is an analysis.
Analysis, right.
So that's what the big focus is now of a lot of computational biology research.
But that's what you're working on.
That's what we work on, right? So,
right? Yeah.
So our raw input is DNA sequence, and our challenge is now to understand what that means.
Right.
Right, and how does it correlate with a clinical phenotype?
There's a promising future actually within your life, maybe not mine, but your
Well,
life at least, maybe. You know.
Well, yes, I'm sure you have a lot. I'm sure you have many more years in which you could see many changes. I mean, I really am very optimistic that there's going to be dramatic changes in the near future. I mean, the fact that we've reached to a point where it only costs $1,000 to sequence one person,
right,
in the space of days. Right. That's a huge.
But see, that is fine. But if you cannot analyze that, certain part of the genes, then it again we are back to square one.
Right. So, but we are making progress.
Okay.
So there's been a number of successes, and it's all really been driven by genetics and the sequencing of the human genome. Had we not sequenced the first draft, our progress would have been very much delayed in interpreting what these genes do. So it's been influential. It hasn't been as fast, our progress, as some people would have liked.
Right.
But there are a lot of rallying calls to see the end of certain diseases in your and my lifetime, particularly in cancer. So the war on cancer was declared by Nixon in the 1970s, and there's been a great deal of frustration. Here we are, 40 plus years later, and we still don't have a good handle on most tumors.
Wow.
But it's only been in the past few years have we able to really understand that cancer really is a genetic disease. So if we understand the genes, understand the DNA, we can get a much better foothold into knocking this thing out in the future.
And there are many types of cancer, so you have to really kind of understand that each one is different.
Well, it's not just that, right? I mean, as you say, you can get tumors of different parts of the body, very different, and they affect different genes.
Right.
But even from person to person, they're very different.
Right, exactly.
I mean, if I had a tumor of the skin, that'd be very different from your tumor of the skin. My melanoma would be very different from your melanoma.
Right.
So there's a great appreciation of the variability, and this is why we need to be able to sequence each individual.
So let me ask you a question. We both are from India. Our origin from India. Like your parents are from India and Pakistan. I'm from India. I was born and brought up in India. How India plays a role in all this thing? What is the research going on in India? We got pretty smart people there.
Right.
So what do you think?
So India has some fantastic world-class research institutions. The IIT institutions, the Indian Institute of Technology. But traditionally, the students who come out of that have been more into the physical sciences, so mathematics, electrical engineering, computer science, and a good deal of them applied to the United States or England or other places to pursue graduate studies in the similar fields. And some of them would actually go off to Silicon Valley and have their own startups and become gazillionaires. So there hasn't been an influx of smart people, smart students from India coming into biology.
Okay.
But I think that's slowly changing now because biology is becoming much more of a computational academic research discipline. We're seeing more and more students from India, from these institutions, trickling into this and applying their trade, their expertise in algorithms and computer science into biology. So I definitely noticed a change in this in the past few years. Within India itself, they don't have a big infrastructure for sequencing, and that's primarily, I think, due to cost. I mean, they're expensive. The best and fastest machines nowadays cost $1 billion, and you have to buy them in bulks of 10. So it's $10 million.
Wow.
Right. So it's not something any institution can buy so easily. You can't write a check for this. And so where India's strengths lie in is actually in the interpretation, the computational biology.
Right.
A lot of this information which we've generated is now publicly available.
Right.
Right. So the Human Genome Project has released the draft of Human Genome online, so anybody around the world can download this. There's been various international collaborations on sequencing lots of different individuals from different parts of the world, and this information is all online. So India doesn't have to invest so much infrastructure in sequencing. What they can leverage is their expertise in analytics.
Analytics, right?
Analytics, and so I definitely see, a big growth in that sector in India. Having said that, I do believe sequencing will become sufficiently cheap, where which they will be able to generate their own data. And that's going to become really important for personalized medicine. I mean, the diseases that may inflict upon a Northern European population or Latin American population are going to be very different at a genetic level from the kind of diseases that you may suffer from in India. So they will have to, if they want to really make big changes into their clinical sciences, they really do have to generate their own data and sequence Indian individuals.
Right, to benefit India, basically.
Exactly.
They have to have their own data, basically. And as you mentioned that, the machines are becoming like an iPhone.
Well, that's the promise. I mean, there's
so that, if that happens, then obviously India will be able to, a lot of machines will be there in India.
Right. And then,
and we have our brain power.
Right. There's no shortage of brains in India.
Exactly.
Right.
Exactly. We've got tremendous brain power.
Right.
So, okay. I mean, that's pretty good. That's promising, actually,
yeah. I mean, I have to say, India has been relatively underrepresented in the genomic space.
Oh, okay.
Right. So far. And I think this is primarily due to the cost and the infrastructure, but that's hopefully will be changing.
With the right leader, it will change probably.
Well, right leader and diminishing costs.
Diminishing costs.
Both
of them.
Right. And India has a different set of clinical issues than most of the world.
Right.
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 There's still a lot of child-borne diseases and ailments that we don't typically suffer here in the United States. But as India progresses economically, it's going to become an aging population. Right. The average age is going to be increasing. Average life expectancy is going to be increasing.
Right.
So the kind of ailments that you suffer at old age are going to be creeping up on the Indian population. So I expect to see diseases that you normally associate with elderly people, like cancer, Alzheimer's. This is going to become a bigger problem in India. So simply because of the economic success that's been that India has incurred recently. So if they want to deal with this, they really have to get into the genomic space.
So Mickey, many of our community is like in medical field, like you wanted to, obviously you are going to be a doctor, but you didn't become a doctor. But there are a lot of people who are in our community who are really a doctor, right? And they are having all the practices all over. What do you see their, change in their practice in, let's say, next 10 years?
Okay. I think there's...
Because of your research, or is combined research rather.
So there's a real fundamental qualitative change, I think, in the practice of medicine. I foresee in my lifetime.
Okay.
Back when I was in medical school, medicine was, for the most part, fairly qualitative. There wasn't much quantification at all. And in fact, it used to be a matter of pride for most doctors to claim they were bad at math. They really hated, the physical sciences. And they're much more enamored with the biological sciences, and that makes sense. I don't see that being tenable in the future. I think the doctors of the future are going to have to be very conversant and very easy to understand and interpret high amounts of data, large amounts of genetic data. So what I see in the future is that doctors are going to have to understand genomics, genetics, far more than they had to previously. They're going to have to be able to deal with interpreting large amounts of data, and it's going to be, I believe, fairly routine for DNA sequencing to play a big role in a doctor's clinic. So I definitely foresee a future whereby if you were a patient, if you were to see a doctor, your doctor would have your DNA sequence available to them, by which they can interpret your condition and prescribe something that was appropriate to you. So there's going to be a real need for doctors to be much more computer savvy than used to be.
Oh yeah.
Right. I mean, that certainly wasn't the case when I was in medical school. Right? Most doctors were not that happy to use computers, and and this has really been driven by a lot of really smart people from the computational sciences going into biological sciences.
Wow.
And in fact, I have to say, some of my best interviewees who I interviewed for a position in my lab, they sometimes have competing offers at places like Facebook and Google, and and it's a very similar job requirements.
Right.
I mean, it's be able to program and analyze large amounts of data, whether it be DNA sequence or social networks, or looking at long lists of names and lists of friends and drawing these graphs, then looking at interactions. Here I'm looking at interactions between genes. So it's very similar, just the
commercial application.
Exactly. It's just very different, the application. And I see that thing being much more integrated in the future.
So I see, happening like there will be large data centers, where they will store everybody's genes, sequence, right? And then if somebody goes into a doctor, he can just go into their, that huge network and find the sequencing or whatever the results are. Is that something? I mean, each doctor cannot store the huge amount of data in their place.
Yeah, so storage is a real problem right now.
Yeah, storage is a real problem.
Storage is a real problem. That's a problem we have to deal with, right? It isn't so bad storing one person's DNA,
right?
But when you're storing large populations, there's a lot of processing power that needs to go into play. To analyze 1 million genomes, you need like 1 billion computers running for an hour, 1 billion CPU hours.
Wow.
So we need to. So these are computational problems we have to contend with, and so we have to develop scalable methods that can contend with this.
Who is the spearheading this, to increase the storage and increase the processing power in terms of the companies I'm talking about, like IBM, I mean, are they playing role in this?
So most computational biology departments at universities and research institutes, they usually have their own large supercomputer.
Right.
So these are lots of computers in parallel working together,
right,
to process large amounts of information. And that's worked out fine for the past few years, but that's not going to work out fine in the future, because the amount of data that's coming in is growing faster than our ability to buy more machines.
Right.
So there's we have to think of other solutions. So one solution is to use the cloud.
Right.
So there are companies like Amazon, Google, who are offering storage space, where you can just offload your data, transmit it by internet. They store it, and they're also offering computational power. So you don't have to buy your own computers. You can just submit your own programs and run it on their machines. And you pay by the hour of the processing time. So that's one possible solution which we'll have to contend with.
Do you use Amazon or anything like that?
I've had experience with Amazon in the past, and it's certainly much cheaper. You don't have to pay for your own IT staff to maintain your own computer. But one of the issues with Amazon is it's still pretty slow to transfer data.
Oh yeah.
It's still transferring large amounts of data. Another issue is privacy.
That's what I was going to ask.
Yeah. So you can, as you can imagine, there's huge amounts of ethical boundaries which we need to discern and figure out what is appropriate.
Right.
Who actually owns your DNA?
Exactly.
Right. And how can people use that information? Against you, if they had to, and so people are naturally cautious about disseminating their DNA. Right. So most clinics go to great pains to anonymize DNA sequences, so that if they have something, some data, they make sure that they try to remove all identifiers so people aren't able to identify if they're working on analytics.
Because I can see a large company, a commercial company, they can easily use this data to do something where they can profit more in the future. Well, if it is available to them, right? I mean,
Yeah, so one hypothetical scenario is that you might imagine an insurance company.
Right.
Right.
Right, exactly.
Using
Yes.
your DNA to decide your premium on you.
Exactly.
And then we have to decide, is that really fair?
Right.
Just because I have this DNA sequence which I'm headed from my mother and father, should I have to pay more than you?
Right.
Right. So that's all those things have to be ironed out, and we haven't quite got there yet. But it's a brave new frontier.
Yeah.
You know, it's a bit like the Wild Wild West, and we don't really know what are the appropriate conditions by which we should proceed.
So there'll be more questions than answer, I guess.
There'll be more questions, right?
Right,
Yeah.
because I mean, it's like the example you gave, insurance premium. Like if insurance company knows somebody doesn't have a good DNA, they may not insure that person.
Yeah, I meant that's a possibility. I mean,
Right.
Right.
So so they will keep on making money and never pay anybody.
Right. I mean, they might treat that as a pre-existing condition, even though you haven't really developed the the ailment as of yet. And this is going to be a problem not just here in the US but elsewhere, because, as I said, DNA sequencing has really come down in price. It's still fairly expensive to buy these machines. It's $1 million, as I said. But there's promise of a new generation of machines, which are going to be no bigger than my iPhone.
Interesting.
So currently these machines are, size of a table, essentially, and they're fairly heavy. But the new generation of machines are going to be portable, faster, and it's really going to democratize genetics.
Wow.
So there's only, a few key places around the world which are major centers for genetic research. But once it becomes cheaper and faster, this is going to be available to anybody in the near future. So the problem of DNA data is only going to get worse. Right?
Yeah, so you have to deal with ethics, you have to deal with ethical privacy, and you also have to deal with negative effects, like people taking advantage of your learning your DNA, things like that.
Right.
There will be the negative effect of it, but then on the other hand, there are a lot of positives also, because then you can be treated very well by certain disease, right?
Yeah. It's interesting from a scientific perspective, from a scientist's perspective, how we deal with these issues. So I try not. So we're not. Obviously we don't think about commercial applications. We just think about, the whole point of what we do is to understand the biological basic principles of disease.
Right.
So that's our motivation for this. But there are possibilities of which this research could be used against other people, and so those are things that lawyers and policymakers have to contend with, right?
And certain countries are more stricter than the other countries. Like China, it will be a different story than the US.
Right. You might imagine that some countries are much more lax about DNA, anonymity, and what you can sequence, what you can't sequence, and what information is public, what information is private. And so that raises a whole host of challenges. If you're transferring information from country to country, you have these international boundaries with different laws.
Exactly.
So how do you deal with that? And that's something we haven't really had to deal with.
So it takes
so far.
It can take a long time, very long time, right? I mean,
but it's inevitable. I mean, it's inevitable. It's coming down the road. I mean, so we're going to have to deal with this. I mean, and faster than we think, right? It was only a few years ago when we only had a few humans sequenced. 5 years ago, we only had a few humans sequenced. Now we have tens of thousands.
Oh, okay.
Tens of thousands.
So,
and there's an initiative in United Kingdom by which they're going to be sequencing 10,000 individuals.
Oh, okay.
Right. And ramping that up to 100,000 individuals.
Oh, yeah.
Yeah. And the idea is that this pilot program would set the guidelines for the future by which they would imagine you would sequence every newborn child. So,
oh, okay.
Yeah. So it is coming down the road. We just have to be able to deal with it. And
right, and the storage issue has to be solved.
The storage has to be solved, and the transmission
has to be solved. Communication.
And really the interpretation.
The interpretation is that's the key, I guess, right?
Right, that's a key thing. Right, that's a real bottleneck.
Right.
I mean,
yeah.
I mean, it's it's a bit like getting a book in a foreign language.
Right.
All the spacing has been taken out, or the punctuation taken out, and you have no idea what this book means.
Right. Yeah.
I mean, that's a bit like the analogy. So so you have to be able to pass that information to see how does it reflect in the underlying biology. Right.
Which country is ahead in this? I mean, in your, like we are now global, right? Everybody's connected to each other, like, compared to.
So the United States has really spearheaded the Human Genome Project, and this was a project that sequenced the first human, and it was completed in 2003. But this is spearheaded by the United States, but it was a collaborative effort with a number of other countries.
Right.
There's a few groups in the United States which can have really risen up as big genome sequencing centers, and what I mean by this is that they have quite a few machines running in parallel. There's Cold Spring Harbor, there's a group in Boston, Massachusetts, there's one in Seattle. There's countries in Europe, for example, England, the Sanger Center in Cambridge is a bigger genomic sequencing facility, and China is really rising up, and they have the largest genome sequencing facility in the world.
Wow. Okay.
So last I heard, they had about 150 machines, running, and they're churning out data,
and running out data every 3 days. One person.
Yeah, exactly.
To 120 people per year.
And so, it's actually faster than that. There's a new generation machines which actually go faster.
Oh, so it goes faster.
So new generation machines can sequence 15 individuals in 3 days.
Oh, okay.
Right. So they are the biggest churner of information. So it really is like a factory. I mean, you just have all these machines in parallel, and there's producing all this data, and there's all these computer scientists. I mean, this certainly wasn't the biology lab that I was growing up in back then. It's all big data, big machines, and a large computational infrastructure.
I guess big data is also going to be useful in commercial applications, right? I mean, because right now everyone holds their own data, and at the end of the day, everybody uses their own data. Basically, I have my own closet, and I use my closet, and you have your own closet. Everybody is combined together, and that's the big data in commercial application, like your example of Target the other day.
Yeah, so big data is routinely used by commercial industry and the finance industry.
Right.
And it's really, it's only when they aggregate data from lots of different individuals can they actually make use of this. And the example is of this story that I read in New York Times. I mean, it's a really interesting story that does the rounds in conferences. A few years ago, there was a gentleman who approached the manager of this retailer, Target, very angrily. And he asked to speak to the manager, and he was complaining why was his daughter, who was only in high school, getting all these coupons for baby clothing, and cribs and such forth. It felt to him almost as if Target was promoting teenage pregnancy.
Right.
And the manager had no idea what he was talking about, and apologized profusely. And the father went back home, and the manager followed up a few days later, and again called to apologize. Didn't know what was happening, and that was an interesting conversation, I believe, because the father, I guess, had since spoken to his daughter, and realized she was pregnant, and apologized to the manager.
Oh my goodness!
So Target, the retailer, knew that his daughter was pregnant even before the father did.
Before the father did.
Even before the father. So how did they do this?
Yeah.
So whenever you go to a big retailer, you buy certain items, and if you have one of those cards, you have an ID associated with you, and they keep track of all your purchases. So there's all these big corporations that are monitoring what are you buying, right? And they have certain information about who you are, male, female, where you live. So certain demographical information about you.
Right.
Now, what's great about this is you have all this data, and then all this historical data, and then as a statistician, you can go back and try to find out patterns.
Right.
So they had a statistician at Target, and he picked up certain things that were useful well, not useful, but kind of interesting. He noticed that, well, he knew when people started buying baby milk, then they obviously had a newborn child.
Right.
But he can also look at what they bought before,
right.
when they were pregnant. And he's noticed certain patterns in their purchases. For example, a lot of women in the second or first trimester would buy lotions that are unscented.
Right.
Or they might be, if they're close to the third trimester, they might be on the lookout for cribs or baby clothing.
Right.
And so he deduced, if he monitors some of those purchases, he'd actually predict which of these customers are going to be having a baby anytime soon. And he can actually narrow it down to a certain small time frame as to when the baby is due. Right. So this is amazing, right?
Amazing.
You have all this historical information, and you can use this. You can use machines to learn something about the individual. This is called machine learning. It's an offshoot of artificial intelligence.
Wow.
Right. You take all this data and you process it in the computer. You develop various algorithms, and what the machine does, the computer does, allows you to predict certain buying habits of individuals in the future. So this is routinely done by corporations. The same thing happens in the finance industry, right? Hedge funds want to predict the price of stocks, right? And the way they do this is by looking at historical data. And so based on historical data, can they predict data in the next millisecond or minute or sometime in the future? And if they have lots of historical data, they can somehow train their algorithms to be much more accurate in their predictions. So like I said, big data has been around for a while. It's only now has biology now become one of the big drivers of the big data industry.
Could it be like different corporation will like put together their data, like for example, rather than using individually their own, that also can come up, right, in later on, that like rather than one financial institution using their own data and predicting their own, rather than combining like all financial industry companies.
Well, I mean, the stock trade data, historical, is all, I believe, publicly available. Right. The only way a hedge fund can outcompete another hedge fund is if they have a better algorithm. Right. So they don't want to share their secret sauce. Right. They don't want to share their magic crystal ball, which they can predict and analyze the information. But the data is certainly available, and you can download that data.
So let me go back to something different subject now, right? You are your parents are from India and Pakistan, right? So obviously, what do you do? Not when you're working is fine, but what do you do outside of work? What is your hobbies? What are your hobbies?
Oh, a professor has no hobbies.
Yeah.
Oh no, this is
in your spare time. What do you do?
In my spare time?
Yeah.
So I live in Manhattan.
Right.
I don't live out here in I work out here on Long Island, so I get to enjoy all the benefits of being in the city. So being close to Central Park, the diversity of people, the energy of people, the different cuisines that you can sample. So that's more than enough to keep me busy. Over there, right?
Oh, okay, okay. Is there any other, like a sport or anything you do, anything at all?
Oh, I used to fence pretty seriously back in England. I haven't done that in a while.
Oh, okay.
I played tennis fairly regularly. I should get back into that. But I do cycle on a regular basis.
Oh yeah.
When the weather's nice nowadays.
Okay. What type of food do you like most?
People ask. So whenever people ask me what's the best food, my canned answer is my mum's. So Indian food. That's what I grew up on.
Indian food, right?
Yeah.
Okay. So, do you go to? You have to go to England, right, to really eat your mom's food.
Yeah.
Right? And how often you go there?
Not as much as I should do, obviously. You should. Yeah. It's probably like once a year now.
Once a year?
No, she comes and visits. So
she comes and visits.
Yeah,
okay. That sounds fine. That's fine.
Right. Yeah. And there's a fair number of Indian restaurants in New York City.
Oh
and,
yeah, a lot.
Yeah. So
right. Yeah.
Each has their own flavor. Right.
And you're single, I guess.
No, I'm actually married.
You're married?
Yeah.
Oh, okay.
Yeah. So I have roots here now.
Oh, okay. Good.
Yeah.
Good. Yeah. Okay. Good. Yeah. What is what do you think of your you're right now working at Cold Spring Harbor, and I understand you're also part of big data, and you're also part of a few big data companies, as with that I remember last time. So what is your future like? What do you think for yourself in 5 years?
I would be. I'm heavily optimistic. With the funding that we have now and our ability to sequence more genomes at a faster rate, we are going to make a much bigger attack on tumor. And cancer, tumors and cancers in general. So cancer is really interesting from a genetic point of view, because when a tumor develops from a single cell to something the size of a golf ball or slightly larger, each of those cells is very different from every other cell. They have different mutations. So you don't want to sequence just one cell. You want to sequence
everything.
100. Well, that's too expensive, right? You have millions of cells, right? You can't do a billion whole genomes. So maybe you want to sample a few. To get a feel of the whole variability. So this is what we're currently doing at Cold Spring Harbor. We're pioneering methods to sequence individual cells from a tumor, hundreds at a time, to explore the entire range of mutations in a tumor. And that's really been quite a revelation to us as to how heterogeneous, how variable the tumors are, even within one patient. And this has really helped us to understand why some patients, when they undergo chemotherapy, the tumor seems to be shrinking, but then it reappears. And what's actually happening is that there are a subset of cells which have mutations that are resistant to chemotherapy. Not every cell is the same. If every cell was the same, the tumor respond exactly in the same manner. So this really speaks to the need for having maybe a cocktail of therapies rather than a single magic bullet. So that's really where we're going for, doing single cell sequencing. Right? And that's a really exciting time, because just think about it. You're taking a single cell, something that's microscopic, taking DNA from a single cell, and do that for many individual cells. So there's a whole range of technology questions which we have to contend with, and interpretation questions.
Any private companies are doing this kind of research, or it's only mostly labs and all that?
There are, so in the DNA sequencing universe, there are biotech companies that are focused on really the interpretation. So they're doing a lot of the analytics, right? So the idea is that you would they would provide some qualitative information as to what this sequence means. There's a company called 23andMe, based out in California, by which you can actually get your certain parts of your DNA sequenced. You take a cheek swab, put it into an envelope, send it out to California, and a few weeks later they tell you, what is your DNA sequence at certain parts of your genome, not entire parts, otherwise it'd be too expensive. But the parts that they look at provide information about your risk for certain disorders and your ancestry, and where you've come from recently, and how much Neanderthal DNA you have mixed within you.
Interesting. 23andMe?
23andMe, yeah.
Okay. Good, very good. I mean, this is a very deep subject, and obviously we have a time limit here. So I think, I really want to thank you for coming and giving us knowledge, which I'm not, knowledgeable in this, but I learned a lot from you the other day and now also. And I hope you can, when you reach any milestone, you can still come back again on the show.
Oh, very happy to.
Yeah. Thank you so much for joining us today. If you have any questions, comments, you can email me at rajamitv@gmail.com. That's again rajamitv@gmail.com. You can watch our prior shows on YouTube at youtube.com/Infosys International. Again, that's youtube.com/Infosys International. Until next time, have a great week.
Thank you.